Recording in progress. Recording in
I believe that now we must work.
Good morning. Am I audible?
Yes, ma'am.
Yes, ma'am.
Okay. Shall we start the class?
Yes, ma'am.
Okay, so how was your sessional exams? I can't hear any response. Was it too easy or was it too difficult? Or was it okay? No response means either it would be too easy or it was too hard. Okay, so you are not interested in responding. Then we'll move on to the class. Okay, in your last class, already a new system has been started, isn't it? We had begun with the introduction to central nervous system. So today we are moving on to the most important part of nervous system, which is your synapse. Now I'll be covering this topic under 3 classes. Today we'll just see the introductory part of synapse. So what is your idea about synapse? Yes, roll number 5. If you're not responding, then I'll mark absent. Roll number 5 present. Roll number 6. Yes, tell me what is your idea about synapse? What do you mean by synapse?
Ma'am.
Yes.
Hello, ma'am.
Yes, I can hear you.
Hello.
Yes. What do you think is synapse? Have you heard of this term? Can you hear me? Roll number 7.
Yes, ma'am.
Yes, tell me,
Hello.
what is your idea about synapse?
Junction between two neurons.
Yes, it is a junction between two neurons. We have already seen what is neuromuscular junction, isn't it? It is a junction between a neuron and a muscle. Whereas synapse means it is a junction between two neurons. Now, if you go through the different functions of the brain, you can see that brain is performing complex variety of functions, isn't it? If you go through your day-to-day activities, the voluntary activities that you carry out, the involuntary activities that are being carried out, the learning process which you go through, memory, everything is related to your nervous system. Now, have you ever wondered how brain is performing all these complex functions? It is mainly with the help of a number of signals that are being transmitted throughout the different parts of the nervous system. And the basic medium for the transmission of these impulses is through the synapse that is present between the different neurons. You have already seen that the neuron is the structural and functional unit of the nervous system. And for the signals or impulses to travel from one neuron to another neuron, you require a medium. And the synapse acts as a medium for the passage of the information through different parts of the nervous system. Now, the synapse, it is not an anatomical connection between two adjacent neurons, but it often acts as a functional. Only a functional connection is present between two neurons that you call as synapse. So on that note, we'll begin the class. So the specific learning objectives include, you need to know the definition and types of synapse, functional anatomy of a synapse, then the different steps involved in the synaptic transmission, then what are the different types of synaptic potentials, then you need to know the properties of synapse and synaptic inhibition. And finally, few points regarding neurotransmitters. So these are the headings that I will be covering in the 3 hours allotted for Synapse. So today we'll just begin with the introductory portion of Synapse. So as I have told you, synapse is a functional junction between two neurons. There is no anatomical continuity between two neurons. Only a functional junction is between two neurons, and that is called as synapse. Now, the term synapse was coined by Sherrington, and it means to clasp. Okay, the word meaning is to clasp. And synapse, it is known as the most important single determinant of the CNS function. Now, to define synapse, it is defined as the junction where an axon or some other portion of one cell, that is one neuronal cell, terminates on the dendrite, soma, or axon of another neuron. Or in some cases, it could be a muscle or gland. I hope you can recall the terms dendrite, soma, axon, etc. All these are parts of a neuron. So to define synapse, it is the junction where axon or some other portion of one cell terminates on dendrite, soma, or axon of another neuron, or in some cases, it could be a muscle or gland. If it is a muscle, you call that junction as neuromuscular junction. Okay, so now we'll briefly see what are the main functions of this synapse. So the most important function is transmission of impulses throughout the different parts of the nervous system. Now, some synapses has also got the ability to block or inhibit impulses, while some synapses can turn a single impulse into repetitive impulses. Also, it can help to modify impulses. So it helps in the transmission of impulses, blocking or inhibiting impulses, changing an impulse to repetitive impulses, and in the modification of impulses. Finally, the synapse can also help to integrate one impulse with another. So with the help of all these functions, synapse will help to transmit the impulses through different parts of the nervous system. Now, you can classify neurons based on the synapse made into a presynaptic neuron and a postsynaptic neuron. So what do you mean by presynaptic neuron? Presynaptic neuron means a neuron which makes contact with the other neuron. That is, in a presynaptic neuron, the impulses will be conducted towards the neuron. And postsynaptic neuron means it is a neuron with which the synapse is being made. Okay, so there the impulses will be conducted away from the neuron. So if you consider two neurons A and B, and the impulse is traveling from A to B, then A is regarded as the presynaptic neuron and B is regarded as the postsynaptic neuron. Okay, neuron which makes a connection with another neuron is called presynaptic neuron, and the neuron with which the connection is being made is called the postsynaptic neuron. Is that clear?
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
We'll move on to the different types of synapse. So you can classify synapse based on its structure as well as based on its function. So based on the structure, you can classify the synapse into axodendritic synapse, axosomatic synapse, axonic synapse, and dendrodendritic synapse. So from the word meaning itself, you can describe each type of synapse. Axodendritic means it is a synapse between the axon of one neuron and the dendrite of another neuron. Axosomatic means the synapse between the axon of one neuron and soma or the cell body of another neuron. While axonic means it is a synapse between an axon of one neuron and an axon of another neuron. And dendrodendritic is between the synapse between a dendrite of one neuron and a dendrite of another neuron. So this is the structural classification. Then you have got the functional classification, that is based on the function performed by the synapse. You have got chemical synapse, electrical synapse, and conjoined synapse. And then there is another third variety known as reciprocal synapse. So we'll see each one. Coming to the structural classification. So you can see the diagram of the different types of synapse based on its structure. So this is the axodendritic synapse, that is the synapse made between the axon of one neuron and the dendrite of another neuron. So this axodendritic synapse is the most common variety. About 80 to 90% of the synapse present in the nervous system is mainly axodendritic. Okay, so here axon of the presynaptic neuron makes a connection with the dendrite of the postsynaptic neuron. Now the second one is the axosomatic. You can see here the axon of the presynaptic neuron is making a connection with the cell body or the soma of the postsynaptic neuron. Then third one is the axoaxonic, where the axon of presynaptic neuron is making a connection with the axon of postsynaptic neuron. This is the least common among the four types of synapse, axoaxonic. And then you have got the dendrodendritic synapse, where the dendrite of the presynaptic neuron makes a connection with the dendrite of the postsynaptic neuron. Now, dendrodendritic type of synapse is usually seen in olfactory neurons, that is, neurons that are concerned with your sensation of smell. So these are the different types of synapse based on structural classification: axodendritic synapse, axosomatic synapse, axoaxonic synapse, and dendrodendritic synapse. Axodendritic synapse is the most common among these four. Now coming to the functional classification, based on the function performed by the synapse, you have got a chemical synapse and an electrical synapse. Now, chemical synapse means here the synaptic transmission will be carried out with the help of a neurotransmitter. Okay, just like the transmission occurring in the neuromuscular junction, in chemical synapse also a neurotransmitter will be involved. The presynaptic neuron will be releasing neurotransmitter in response to a signal that is reaching the presynaptic neuron, and it will discharge the neurotransmitters, which will go and bind to the receptors, specific receptors that are present on the postsynaptic neuron. So a transmission which is involving a neurotransmitter is called as a chemical synapse. Now this is the most common type of synapse based on the function. Now here the conduction of impulses will be always unidirectional, that is from the presynaptic neuron to the postsynaptic neuron. Now coming to the electrical synapse, here the presynaptic neuron and postsynaptic neuron are lying close to each other. As you can see in this figure, they're lying very close to each other and they form gap junctions between adjacent neurons. Okay, so that is electrical synapse. You have got presynaptic and postsynaptic neurons lying close to each other and they are connected by gap junctions. So here the transmission of impulses is bidirectional. That is, impulses can travel in both directions through these gap junctions. Now, another difference between the chemical synapse and electrical synapse is that in chemical synapse, there is a phenomenon called as synaptic delay. That is, there is a certain amount of time is required for the impulses to travel from the presynaptic neuron through to the postsynaptic neuron. So synaptic delay is present in case of chemical synapse, whereas in electrical synapse, since both the neurons are lying close to each other and are connected by gap junctions, there is no synaptic delay. So transmission of impulses is much faster in case of electrical synapse. So these are the basic features of chemical synapse and electrical synapse. I'll repeat once more. Chemical synapse always involves a neurotransmitter. And here the conduction of impulses will be slower because there is a synaptic delay for the transmission of impulses from presynaptic neuron to postsynaptic neuron. Whereas in electrical synapse, there is no synaptic delay. The conduction is bidirectional and it is faster than the chemical synapse. Now there is a third variety called as conjoined synapse, where both chemical synapse as well as electrical synapse can occur in the same synapse itself. In some instances, there can be chemical synapse, whereas in other instances, there can be electrical synapse. So if both chemical synapse and electrical synapse are involved in a simultaneously in a synapse, you call it as conjoined synapse. So that is regarding functional classification of synapse. So we have covered anatomical classification and functional classification. Now there is something called as reciprocal synapse. Reciprocal synapse means normally I have told you there is a presynaptic neuron and a postsynaptic neuron, and impulses will travel from presynaptic neuron to postsynaptic neuron. Also, in case of a chemical synapse, as you can see in this figure, the neurotransmitter will be involved. It is released from the presynaptic neuron and it will bind to the receptors present on the postsynaptic neuron. So here, the receptors are present only on the postsynaptic neuron. But in case of reciprocal synapse, both the presynaptic neuron as well as the postsynaptic neuron will be having the receptors for neurotransmitters, as well as neurotransmitters will also be present on both the presynaptic as well as postsynaptic neuron. So in reciprocal synapse also, the synaptic transmission can be bidirectional. One example is again the synapse that is seen in your olfactory neurons, or the neurons involved in the physiology of smell. So you will learn in detail regarding the olfactory neurons when the physiology of olfaction is being taken. Okay. So this is just an example of an olfactory neuron. It has got different cells called as mitral or tufted cells. And the basic synaptic mechanism that is occurring is these mitral cells, they can stimulate another type of cells called as granule cells, whereas this granule cells in turn can inhibit these mitral cells. This occurs mainly due to the presence of reciprocal synapse, which is being involved here. Now, just remember in reciprocal synapse, the receptors for neurotransmitters are present on both the presynaptic neuron and postsynaptic neuron. Also, the neurotransmitters are present on the both presynaptic and postsynaptic neuron. So the new synaptic transmission will be bidirectional. And an example is the synapse that is seen in olfactory neurons. Now, the mechanism of how this reciprocal synapse is being taking will be dealt in the physiology of olfaction. So that is regarding the types of classification of synapse. We have seen the structural classification and functional classification. Now we'll move on to the functional anatomy of synapse. You need to know the functional anatomy of a chemical synapse. So if you remember, recall your class on neuromuscular junction, I've already taught you about the anatomy of a neuromuscular junction. Now, the anatomy of a chemical synapse is also more or less similar to that of a neuromuscular junction. The difference is that here we are going to study the synapse between two neurons, whereas in neuromuscular junction, there is a connection between a nerve or a neuron and a muscle. So coming to the parts of a chemical synapse. So can you tell me what is terminal button? We have come across the same terminology in neuromuscular junction. What is synaptic knob or terminal button?
The end of the presynaptic neuron.
Yes, it is the terminal portion of the presynaptic neuron. Okay, what does it contain? Contains the neurotransmitters present within the synaptic vesicle. So this is the enlarged portion of a synaptic knob or terminal button. So you can see it contains mainly mitochondria, synaptic vesicles containing the neurotransmitters, and few lysosomes. So mitochondria, the function of mitochondria is to provide the energy for the release of neurotransmitters from these synaptic vesicles. Okay, so that is regarding synaptic knob or terminal button. Now a few points regarding synaptic vesicles. Now, synaptic vesicles, they are of different types. You can have round or spherical clear vesicles. Now, usually such vesicles will contain excitatory neurotransmitters like acetylcholine. If you remember neuromuscular junction, acetylcholine was a neurotransmitter involved in neuromuscular transmission. So, such neurotransmitters are mainly present in round or spherical clear vesicles. Then you have got another variety, that is flat clear vesicles. These vesicles usually contain inhibitory neurotransmitters. Can you name one inhibitory neurotransmitter?
GABA.
Yes, very good. GABA is an example of inhibitory neurotransmitter. GABA, glycine are all examples of inhibitory neurotransmitters. Then the third variety is small dense core vesicles. Such vesicles usually contain catecholamines like epinephrine, norepinephrine, etc. And the fourth variety is large dense core vesicles, which usually contain neuropeptides like endorphins, enkephalins, etc. So based on the type of neurotransmitter present within the vesicle, you can have different types of vesicles. Okay. There is round or spherical clear vesicles which mainly contain excitatory neurotransmitters like acetylcholine and glutamate. Then you have flat clear vesicles which contain inhibitory neurotransmitters like GABA and glycine. Thirdly, you have got small dense core vesicles which contain catecholamines like epinephrine and norepinephrine. And finally, you have got large dense core vesicles which contain endorphins, enkephalins, etc. So that's regarding the synaptic knob or terminal button. Now we'll see the next structure, which is the presynaptic membrane. Now, presynaptic membrane is a portion of this terminal button where the synaptic transmission is occurring. So if you see this diagram, this is a diagram of a presynaptic neuron, and this portion is your terminal button. Now, here you can see that there is an area of the terminal button which has been modified to form the area where synaptic transmission is going to occur. And this area is known as the active zone. Again, the term active zone has also come across in neuromuscular junction. So presynaptic membrane, it is a part of the axonal terminal of the presynaptic neuron, or it is a membrane that is lining the synaptic knob. Okay, and it is at this area that the neurotransmitters will be released from the presynaptic neuron. So it mainly contains zones of dense cytoplasm, which will allow the vesicles, which allows organized channeling of vesicles to the presynaptic membrane. So this area, it contains zones of dense cytoplasm. So here these synaptic vesicles will come and organize in such a way that they can release the neurotransmitters into the synaptic cleft. Synaptic cleft is the gap between presynaptic and postsynaptic neuron. So presynaptic membrane is an axonal membrane that is lining the synaptic knobs. It contains zones of dense cytoplasm, which allows organized channeling of vesicles to the presynaptic membrane at a site opposite to the receptors present on the postsynaptic membrane. An active zone or dense bars are the areas where these synaptic vesicles will be releasing the neurotransmitters. Okay. So we have seen the terminal button, presynaptic membrane, which contains the active zone or dense bars. Now the next structure is the synaptic cleft. So as you can see in this figure, synaptic cleft is the gap between the synaptic knob or the presynaptic neuron and the postsynaptic neuron. So the gap is known as synaptic cleft. So it is a gap between the presynaptic and postsynaptic membrane. Normally it is about 20 nanometers wide. So whatever neurotransmitter that is being released from the synaptic vesicles present in the presynaptic neuron has to diffuse through these synaptic cleft. Only after diffusion through this synaptic cleft can it bind to the receptors present on the postsynaptic membrane. Okay, so this gap is about 20 nanometers. And the neurotransmitter released from the presynaptic membrane diffuse across the synaptic cleft and reaches the postsynaptic receptors present on the postsynaptic membrane. Now coming to the postsynaptic membrane. We have already seen that the postsynaptic membrane is the one which is containing the receptors for neurotransmitters that are released from the presynaptic membrane. So postsynaptic membrane lines the postsynaptic neuron or the postsynaptic process. Now, in postsynaptic membrane, there are certain areas that are modified for synaptic transmission. These areas are known by different terms like postsynaptic density or synaptic web or palisades. All these terms are the terms given for the area over postsynaptic membrane that is modified for synaptic transmission. Now it is this postsynaptic density that is containing the receptors for neurotransmitters. And these receptors, they have a characteristic feature. They are proteins in nature. And so they have got two components. They have a binding component and an ionophore component. Binding component means it is the component that is projecting towards the synaptic cleft. And it is onto this binding component that the neurotransmitter will come and bind. Whereas ionophore component is the component that is projecting towards the cytoplasm inside the postsynaptic membrane. So if you see this diagram, you can see these are the postsynaptic receptors. It has got a binding component onto which the neurotransmitters released from the presynaptic membrane is coming and binding. And inside you have got an ionophore component, which is diffusing into the cytoplasm of the postsynaptic membrane. So this diagram summarizes the different parts of a chemical synapse. you have got a terminal button which contains various synaptic vesicles with neurotransmitters. Then you have got a presynaptic membrane that is lining the terminal button, and the area on the presynaptic membrane which is modified for synaptic transmission is called as active zone. Then following the presynaptic membrane, you have got the synaptic cleft, which is the gap between the presynaptic and postsynaptic neuron. And then you have got the postsynaptic membrane, on which you have got postsynaptic density or synaptic web or palisades, which are areas present on the postsynaptic membrane that is modified for synaptic transmission. You can be asked to describe the functional anatomy of a chemical synapse as a part of your essay. So you need to know the diagram and the different parts of a chemical synapse. Now, there is something called as neurexins. Now, neurexins are nothing but these are proteins that can hold this presynaptic and postsynaptic membranes together at the synapse. I've already told you there is no anatomical continuity between the two presynaptic and postsynaptic neurons. So how is this presynaptic and postsynaptic neurons held intact? It is with the help of proteins called as neurexins. They will help to hold the presynaptic and postsynaptic membranes together at the synapse. So you can say that neurexins will provide structural stability to the synaptic architecture. So that is about neurexins. So now moving on to the most important part of today's class, that is synaptic transmission. So the steps of synaptic transmission are more or less similar to the steps of neuromuscular transmission. So you can divide the steps into presynaptic mechanisms and postsynaptic mechanisms. Presynaptic mechanisms means the mechanisms that are occurring within the presynaptic neuron, and postsynaptic mechanisms involve the mechanisms that are occurring in the postsynaptic neuron. So synaptic vesicles, we already know that it contains neurotransmitters, and these synaptic vesicles that contain neurotransmitters are mainly concentrated at the region of active zone present over the presynaptic axon terminal. So the first step is these synaptic vesicles, they will undergo docking and priming. Now, what do you mean by docking? Docking is nothing but it is a process by which the vesicles, these synaptic vesicles, will come and attach to the presynaptic membrane. That phenomenon is called as docking. And priming means it is the process by which the vesicles become ready to discharge their contents in response to a stimulus. So until a stimulus comes, this synaptic vesicles will be in a stage of priming. So the first step in synaptic transmission is docking and priming of the synaptic vesicles. So docking means it is the mechanism by which the vesicles will come and attach to the presynaptic membrane, and priming is the process by which vesicles become ready to discharge their contents in response to a stimulus. So now we'll see what happens when an action potential is reaching the terminal button or the synaptic node. What happens when an action potential is reaching the neuron in case of a neuromuscular junction? Can you recall the steps of neuromuscular transmission? Yes, one of you, can you please tell me the steps involved in neuromuscular transmission?
Depolarization occurs through the T-tubule and
No, I'm
dihydroxyl
asking about neuromuscular transmission. What you are telling is about the mechanism involved in muscle contraction. What happens when an impulse is reaching the neuron, axon terminal of a neuron? What is
Voltage
happening?
calcium channel open.
Calcium channels
Yes,
are open
calcium
due to
channels
depolarization.
open up. And then what happens?
Depolarization.
Hmm.
Release
What do they release?
neurotransmitter.
Yes, in case of neuromuscular transmission, it is the acetylcholine that is being released. And then what happens?
That binds to the
The postsynaptic membrane.
No, in case of neuromuscular transmission, it will go and
Bind the muscle fibers.
yes, muscle fibers. It will diffuse through the sarcoplasm, travel along the muscle fibers, and then your mechanism of excitation and contraction of the muscle will be initiated. Now here, the mechanism is more or less similar. So first of all, an action potential will reach the synaptic knob and it will depolarize the presynaptic membrane and opens up the voltage gated calcium channels. So calcium will diffuse into the presynaptic neuron. Now, what does this calcium do? It will cause exocytosis of the synaptic vesicles. So calcium mediated exocytosis occurs and the vesicles will slowly fuse with the presynaptic membrane. And then they will discharge the contents into the synaptic cleft. Okay. Action potential reaches the synaptic knob. It depolarizes the presynaptic membrane, causing opening up of voltage gated calcium channels and calcium influx. This causes calcium mediated exocytosis of the vesicles. Exocytosis means the contents will be discharged out of the vesicle and the vesicle fuses with the presynaptic membrane and cause the discharge of contents into the synaptic cleft. So this type of discharging the contents is known as kiss and run discharge. They will come touch the presynaptic membrane and then we will discharge their contents and then they will leave the site. Okay. So this type of discharge is called as kiss and run discharge. Now, from the moment of calcium influx up to the release of neurotransmitters, it takes about 200 microseconds. Okay. So this figure summarizes the different mechanisms, presynaptic mechanisms. So here, if you see one, you can see an action potential is reaching the terminal button or synaptic knob. It is causing the opening up of voltage gated calcium channels. This calcium will enter into the cell and it will cause the vesicles to move towards the presynaptic membrane, which is known as your docking process. And then priming occurs, the vesicles become ready to discharge their contents, which is the neurotransmitter, into the synaptic cleft. So until here comprises the presynaptic mechanisms. Okay. Now you need to know about few proteins that are involved at the level of presynaptic processes. One is synapsins. Synapsins, they are nothing but they are a group of proteins that will help the vesicles to remain attached to the cytoskeleton of the nerve terminal. They are a group of proteins that will help the vesicles to attach with the presynaptic membrane of the presynaptic neurons. Okay. Now there is another group of proteins called as RAB proteins or RAB3. Now this is a small GTPase protein that will help in guiding the vesicles towards their releasing site. Now all these proteins, they may be sometimes asked for your theory viva. What are synapsins, what is RAB protein, etc. So I'm just mentioning each one. So RAB protein is a small protein that will help in guiding the vesicles towards the releasing site. Then you have got another type of protein called as snare proteins. Snare proteins are examples of integral membrane proteins that will help to bind the synaptic vesicles with the presynaptic membrane. That is, it is mainly helping in the docking mechanism. So synapsins means there are a group of proteins that will help the vesicles to attach to the cytoskeleton of the nerve terminal. RAP proteins are proteins that will guide the vesicles to the releasing site. Example is RAP3 protein, and SNARE proteins are integral membrane proteins that will bind the synaptic vesicles to the presynaptic membrane. So examples of SNARE proteins are synaptobrevin and syntaxin. Synaptobrevin and syntaxin are examples for SNARE proteins. Now the SNARE protein which is present in the vesicular membrane is nothing but the membrane that is lining the vesicle. So the SNARE protein which is present in the vesicular membrane is called as synaptobrevin, and the SNARE protein that is present in the presynaptic membrane is called as syntaxin. So together they form the SNARE proteins and help in binding the synaptic vesicle to the presynaptic membrane. So we have seen what are synapsins, what is RAP protein, and what are SNARE proteins. So now what is the importance of this synaptic transmission or the presynaptic mechanisms? One importance is in tetanus toxin. Have you heard of tetanus toxoid injection?
Yes, ma'am.
Yes, what is it used for?
To prevent tetany after injuries.
Yes. So tetanus toxin is a neurotoxin that can be released at the site of synaptic transmission. It can prevent this attachment of this synaptobrevin and syntaxins, which I have mentioned a little earlier. So what happens when they inhibit or when these toxins bind to the synaptobrevins and syntaxins? They will prevent the release of neurotransmitters into the synaptic cleft. So synaptic transmission will not occur. So that is the reason why you get spastic paralysis and lockjaw following tetany. So that is one clinical significance. Another example is botulinum toxin. Now, botulinum toxin can cause a disease called as botulism, which is usually seen following ingestion of any type of contaminated food. So there is a bacteria known as Clostridium botulinum that can produce this toxin called as botulinum toxin. And the classical feature that you can get is flaccid paralysis. Here also, what happens is this toxin will go and bind with the SNARE proteins like synaptobrevin and syntaxin, and they will prevent the release of neurotransmitters into the synaptic cleft. So again, here you can get flaccid type of paralysis. Now, spastic paralysis, flaccid paralysis may be new terms to you all, but you'll come across these terms again when the motor system is being taken. So now just remember the few clinical importances related to the presynaptic mechanisms. Now there is something called as Botox injection. Have you heard of it?
Yes, ma'am.
Yes, what is it?
It is used for cosmetic purposes.
Yes, what does it do? Why do you take this injection?
It prevents wrinkle formation.
Yes. Now this is one thing that is becoming popular in the field of cosmetic. So here you can see this injection is mainly used for the treatment of muscle hyperactivity. One use is you can inject this into your facial muscles. It will help to remove the wrinkles present over your facial muscles. So this is widely used in the by the film industry people for cosmetic purposes. So that is one use of Botox injection. Other uses, it can also be used for treating other conditions like esophageal hyperactivities. Any area where you are getting a muscle hyperactivity, you can give this injection. Now it will help you to reduce the hyperactivity associated with this phenomenon. Then other uses include, it can be used to reduce the spasticity that you see with cerebral palsy and all. So all these are examples of clinical significances related to the presynaptic mechanisms. Again, this can be asked for your viva. Examples for presynaptic mechanisms. One is tetanus toxin. Then you have got botulinum toxin. So that is regarding the presynaptic mechanisms. Now moving on to the postsynaptic mechanisms. So what happens in the postsynaptic membrane? The neurotransmitter that has been released from the presynaptic membrane diffuses through the synaptic cleft, and it will come and bind to the receptors present in the postsynaptic membrane. Now, once the neurotransmitter binds with the receptors present in the postsynaptic membrane, conformational changes will occur in the receptors. So what can happen is, two things can happen. Either depolarization or hyperpolarization can occur. Now, when conformational change occurs within the receptors, this in turn will lead to the opening up of ion channels present on the postsynaptic membrane. So when the ion channels open up, there will be diffusion of ions into the postsynaptic membrane, just like the mechanism occurring in your neuromuscular junction. So this diffusion of ions can lead on to the development of potentials within the postsynaptic membrane, which you term as postsynaptic potential changes. Now, based on whether this potential change is occurring towards depolarization or towards hyperpolarization, you can have different types of postsynaptic potentials. So that is the end mechanism in synaptic transmission. So an action potential that has originated in the axon terminal travels across the presynaptic, diffuses across the presynaptic membrane, causing the release of neurotransmitters from the synaptic vesicle. The neurotransmitter diffuses through the synaptic cleft, reaches the receptors present on the postsynaptic membrane. The receptors will undergo conformation present on the postsynaptic membrane. And through these ion channels, ions will diffuse into the postsynaptic membrane. And these ions can lead on to potential changes within the postsynaptic membrane, which are called as postsynaptic potential changes. Now, based on whether it is towards depolarization or towards hyperpolarization, potential changes can be classified as excitatory potential changes and inhibitory potential changes. Now you will be discussing about in detail about the synaptic potentials in our next class. So any doubts in the regarding the steps of synaptic transmission? So today we have covered what is synapse. We have seen the different types of synapse. Based on its structure and function, we have seen the classification of synapse. Then we have seen the functional anatomy of a chemical synapse. The different parts include a synaptic knob or terminal button, presynaptic membrane. Then we have seen the synaptic cleft and the postsynaptic membrane. And I have summarized the different steps involved in synaptic transmission. You have got presynaptic mechanisms like docking and priming. Then we have seen the postsynaptic mechanisms where the receptors will undergo conformational changes. Also, we have seen the clinical importance of synaptic transmission. So any doubts? Shall we stop here? We'll continue with the synaptic potentials in the next class.
Okay.
Any doubts?
Thank you.
Okay, then thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you.
Recording